Okay, actually, normally I like to walk around when I'm talking, but obviously I'm not going to be able to do that. I guess you can all hear me at the back, yeah? Okay. Joe walked. Okay, so this talk is about some compounds called tunicomycins. Actually, it's translocase 1 inhibitors and the target bacterial cell walls and mammalian end glycoproteins. So, for this audience, I hardly need to introduce this subject. This is peptidoglycan. So peptidoglycan is this beta-1,4 linked carbohydrate protein complex that's found in bacterial cell walls. Gram-negative bacteria, it's found between the two membranes. So this is the cytoplasmic membrane and the outer membrane. And we have in here some yellow peptidoglycan. And on the gram-positive bacteria, there's a much thicker layer also outside of the cell. So peptidoglycan is outside of the cells of bacteria. It's a structural feature, and it's totally necessary for the bacteria to survive. You can see it here in an electron micrograph. The membrane just runs through there, and this is the peptidoglycan in a gram-positive bacteria. The structure looks like this, and the biosynthesis, of course, starts inside of the cytoplasm of the bacteria. It's the domain of these genes, these proteins called the MUR proteins. And what the MUR proteins are actually involved in is they take UDP-GluTNAC, UDP-N-acetylglucosamine, through to UDP-MURNAC pentapeptide. So a lot of them are peptidyl transferases. This all happens in the cytoplasm. The step I'm interested in, the part I'm going to tell you about today, is the transfer of this sugar nucleotide onto a lipid carrier called undecaprenyl phosphate, and that's embedded in the membrane here so that we end up with a membrane-bound sugar head group. And basically this again then gets modified again, so we get a disaccharide forming, which again gets transferred outside of the cell. Here we have cross-linking, and this leads to mature peptidoglycan. Now obviously this is a target for a number of antibiotics. The cross-linking step is inhibited by penicillin. Vancomycin inhibits the dealanine displacement steps up here. One of the things I've been interested in for a number of years, probably too many years even, but for a number of years, is this protein here called MRAY. It's also known as translocase 1. And it's a target for this natural product called tunicomycin. Okay, so this basically shows the same thing a little bit, but I just want to show you a little bit about the mechanism. So UDP-GluTNAC gets transformed into UDP-MURNAC pentapeptide, and then this gets transferred onto this undecaprenyl phosphate carrier. All right, so we have undecaprenyl phosphate in the membrane. This is our UDP-MURNAC over here. So UMP is the leaving group for that reaction, and the sugar phosphate forms a pyrophosphate bond to this decaprenyl, which is embedded in the membrane. Okay, this is a very busy slide, but I just want to give you a general idea. I'm going to talk a little bit more about this in a second. This is just to introduce the subject. These proteins, the MRAY, the translocase, they're actually transmembrane proteins. This one has, generally, they have 10 transmembrane domains. That means they have 5 loops that are in the cytoplasm of the bacteria. The active site is actually here, which is this aspartic acid residue. It actually makes an acyl enzyme intermediate. We don't really have time to talk about that. There's a cation binding site over here that coordinates to the substrate. What I want to talk to you, though, is about this loop over here. All of this machinery in here is conserved. This is the catalytic site, so all the family members have the same catalytic site. What I want to talk to you about is this cytoloop 5 over here. And a few years ago, Matt Anderson and I, and also Vavalno's group in Canada, noticed something quite interesting, and that is this loop 5, which is between transmembrane 9 and transmembrane 10, actually has consensus loops within it that correspond to the sugar nucleotide usage by the translocase. Okay, so there are certain family members of the translocase. The MRAY family uses UDP-MURNAC pentapeptide as the substrate. The WEC-A family, the bacterial WEC-A family, uses UDP-GluTNAC, D-GluTNAC as the substrate. And then there are other family members, WBCO and WBPL in Pseudomonas. and these use UDP futanach as the substrate. So by which I mean this is the sugar which is actually transferred as the sugar phosphate to the undecaprenol. Okay, and we notice that there's highly conserved structural features, but they are family specific. Okay, so the MRAY has this motif, the WEC-A has this motif, and the WBCO has this Yershey structure, and this corresponds to the sugar nucleotide that's used as a substrate in the enzyme reaction. Okay, let me tell you about tunicamycin. Everybody should know about tunicamycin. So, starting here, this is the transition state for the MRAY WEC-A translocase family. So this uracil, ribose, and up here is N-acetylglucosamine, or it could be N-acetylmurinac. This is the substrate this is the sugar that's being transferred, the sugar phosphate, dolichol phosphate coming in and displacing that, okay, displacing UMP. Now you can see that tunicamycin looks quite a lot like that structure in a sense, you know, at a first glance, that this is the uracil, and this up here is glutanac, and for tunicamycin that sugar is always N-acetylglucosamine. But this structure in the center here is quite interesting. This is actually an 11 carbon sugar. So this is carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. And it has, you can see it's a dialdose here. It has an N-glycosidic linkage to the uracil, and then it has an O-glycosidic linkage to this glutanac, a 1-to-1 glycosidic linkage, a little like trehalose. Okay, so a little while later, Lin Xu and Frank Monamy and I published some work about a model for how the tunicamycins inhibit these translocases. And so tunicamycin actually binds to magnesium and a number of other cations as well. And this coordinates inside of the translocase and lines things up for this recognition domain, which is this carbohydrate motif that we were just discussing a few seconds ago. So, okay. So I want to talk a little bit about tunicamycin. I'm going to tell you a little bit about some new compounds we found in my lab within about the last 6 months or so. Okay, you're the first audience to have seen this, so please be gentle with me. Okay, so we found in 2011, in collaboration with Wenqing Chen in Wuhan, and we found the operon which controls biosynthesis of the tunicamycin. So there are 12 ton genes. If you express them, transgenic expression, and we did this in 3 different strains of streptomycetes, and you see that you get killing activity on an agar plate against bacillus there. And we're able to identify each of these by just using, this just shows an HPLC profile. What you're looking at there actually on the HPLC profile is the same compound that differs in the fatty acid chain length. Okay, so we have fatty acid tunicamycin with a 15, 16, 17, 18 fatty acid chain length. Okay, so once we had the open reading frames for these ton genes, we can sort of go into function a little bit. I don't want to go into this. We haven't the time to do this morning. I just want to show you a couple of things, though. One is very interesting. It's this ton B radical SAM protein. And what that does, actually, it puts together this 11 carbon sugar from a 5 carbon uridine and from a 6 carbon sugar, okay? So that's a very important step right there, radical SAM protein. The other one I want to just talk about briefly is this ton D, which is the glycosyl transferase. And I mentioned that there is a 1-to-1 alpha-beta linkage between the tunicamine and the glutanac head group. This ton D is a putative glycosyl transferase that actually does that job. Okay, so the radical SAM protein catalyzes a radical addition reaction. So the radical is generated actually from S-adenosyl methionine, and we then get this uridine 5 radical here, which reacts with this 5,6-ene sugar, and this 5,6-ene sugar is generated from UDP glutanac. Okay, the details of this don't matter for a moment or two. And what happens there is that we're starting to form a new bond there, and that gives us this 11 carbon sugar called tunicamine. Okay, now the ton B is unusual. It's unusual enough that we can actually use that as a probe. And what we did was, we used that as a genomic probe of a library that we've generated at the USDA labs in Peoria. And this is a library of the actinomycetes in the NRRL collection. So this is the largest microbial collection in the world, actually. And we're generating a genomic library for all of the strains in that collection. And out of that, we did a search for this TUN-B, and we were sort of hoping we would be able to identify some other strains that might have these TUN genes. And what we found, in fact, was this is Streptomyces chartreuse, which is sort of my rat strain that we originally identified the genes in. And you can see here we identified 3 other strains, actually 4 other strains. I'm going to tell you about 3 of them right now that have the tunicamycin genes in them. And all of this was based on just searching these databases for this TUN-B gene here. So the ones I told you about, TUN-B and TUN-D. Okay, so we take one of those strains, Streptomyces niger, and we grow it in fermentation. And what this shows you here actually is a MALDI-TOF MS profile for the natural products from that strain. We're looking at a mass range here, you can't see this, but from 800 to 900 mass units. And we're seeing 2 sets of compounds that I've called Q1, Q2, and Q3, and T1, T2, and T3. Okay, and these are all in the same sort of mass range there. What's interesting here is that the Q1, Q2, Q3 differ by 14 mass units, and the same for the T series, they differ by 14 mass units. All right, 14 mass units for those in mass spectrometry usually means a CH2, which usually means chain length in a fatty acid chain. So we just purified these by reverse phase HPLC, and then we looked again by MALDI-TOF, and you can see here the T1, the T2, and the T3 compounds, and the Q1, Q2, and the Q3 compounds, so we can actually purify those. And the other thing which is sort of interesting here, if you look at the Q series and the T series, so they actually differ by 16 mass units. So Q1 and T1, the T1 is 16 mass units larger. And the Q2 and the T2 is 16 mass units larger. So we had this curious effect of 16 mass units difference between the two different sets of peaks here. So we did some Orbitrap here. This is kind of a busy slide. I'll just go through it. This is an Orbitrap mass spectrum. And so this here is Q2, and this is T2. We did it on all of them, of course, and I'm just showing you 2 of them. This is the MS peak here, so the Q2 is 829, and this one is 845. This is the 16 mass unit difference I was telling you about. And you can see that in the MS-MS, after the first fragmentation, we're already down to 624, so the same mass. In other words, the first ionization that happened, my 16 mass units went straight out of the window. So we're seeing 205 and 221, the loss of 16 mass units in the head group. Following on from that, MS3, they're identical. And if we look at MS4, fragmentation, we choose an ion there and just fragment it again. Those are also identical. Now, what that's telling me at this stage is that we have very similar compounds, but they differ in the first fragmentation loss, a neutral loss, and that loss is actually the 16 mass unit difference. So the question then arises, what is the 16 mass unit difference in this sugar head group? Okay, bearing in mind that only the tunicamycins that have ever been found have all got N-acetylglucosamine there. So at that stage, I started to get quite interested and quite excited about what we were doing, and I dreamt up a new name, because that's what I do when I get excited. So at that stage, we decided that this is perhaps a deoxy sugar, so we had to go and synthesize a couple of deoxy sugars. So we used a procedure here, which was published a few years ago by Stephen Hanessian. We took N-acetylglucosamine, and we also took N-acetyl-galnac as well. We went through the bromo sugar, and then we reduced this in order to give us the deoxy sugar. We made a deuterated form, and we also made the non-deuterated form. And then after we peracetylate these, we can just run them on a GC mass spec. What I want to show you is that this is our Q series after acid hydrolysis and peracetylation. In blue there, yeah, you can see it's overlapping. That's the main thing. You can't actually see it, but this is overlapping with the quinnac control, whereas the defucnac control is here. And if we look at the tunicamycins that we isolated also from that same strain, from that Streptomyces niger strain, you can see that here the sugar that's hydrolyzed off of there is actually glutnac, and here is galnac just as the other control. And down below there is the mass spectra showing you that it's quinnac. So it looks like then there is a deoxy, a 2-N-acetyl 6-deoxy sugar, and it looks like it's a quinac rather than futac. Okay, and this is just some mass spectra, some NMR spectra. This is a toxy, and this is an HSQC. So actually, since we purified the compounds, we got a complete suite of NMR spectra on this as well. And I want to just show you a couple of things on here which are actually crucial to this, okay? So you can't see the numbers here too well, I'm afraid, but this one here, number 1 here, what that is an alpha 1' anomer. Okay, so this is this alpha beta anomer to anomer linkage. That's the anomeric linkage of that quinavosamine to the tunicamine. So that very much proves that the quinavosamycins have quinac in the head group instead of glutnac. Right? The other key thing here, out of this, the key take-home message, is this little fella up here, which is the 6' methyl group. And that's again because the deoxy sugar, we expect to see that 6 methyl group there, and that's that one up in that position up in there. Right? So we have a pretty good characterization of this compound. This work is not yet published. Okay, so that led to some quite interesting discussion in my lab, actually, about what these compounds are for and why we're finding these compounds and what they might be useful for as well. So we went back and we started thinking about the biosynthetic pathway I introduced to you at the beginning of the talk, and we said that UDP glutnac gets converted into this 5,6-ene sugar. Okay, so UDP glutnac 5,6-ene, and that's, at least hypothetically, that's catalyzed by this protein here called TUN-A. That's not been shown enzymatically, that's just based on homology. And you can see that that ene sugar, if there was a reductase function present, the reduction of that would actually lead to the UDP quinac sugar nucleotide. This then gets epimerized from the gluc to the gal, so we have 4 epimerization of that sugar under the TUN-F. And then we have this TUN-B, which we discussed before, which is a radical SAM protein, which is putting together this 11-carbon backbone from this position here to this position over here. So that's generating our 11-carbon backbone over there. So the interesting thing here is this is a conserved mechanism of biosynthesis in the Niger strain, conserved with the chartreuse strains, clavigularis strains, which also make tunicamycins. And then if possibly there was a reductase step, we could see that that could lead to quinac. And then if we had the TUN-D glycosyl transferase, okay, the glycosyl transferase adds this sugar head group, so we have this 1,11 linkage to this position here, the 11 position. Okay, so now we have tunicamycins, which have the glutnac head group, and we have the quinavosamycins, which have this quinac head group. Okay? All right, so then the other question is, of course, well, what are these compounds for anyhow? Are they any good for anything in any case? Now, the tunicamycins, people have used for a very long time as a tool. Very often it's used to study bacterial peptidoglycan biosynthesis. It's also used very much so because it blocks protein glycosylation, N-glycosylation in eukaryotic cells. So the step that transfers the glutnac onto the dolichol phosphate is the step that's actually inhibited by the tunicamycins. Now that's a very selective step that uses glutnac as the sugar that's the first sugar to go onto the dolichol. So I want to show you this data. I think this is kind of an interesting piece of data. So what we did here is, these are just, you're looking at agar plates, which we've overlaid with a microorganism. So in this case, it's Bacillus. This one is a Pseudomonas, and this over here is a yeast strain. This is just the control, DMSO. Oh, apparently it's methanol, excuse me. And here is commercial tunicamycin. T is commercial tunicamycin. This one here is the tunicamycin that's actually isolated from the Streptomyces niger strain. So that's a positive control, if you like, for the experiments that we'd done earlier. And then this one, Q, is the quinavosamycins. Now, when I initially did that, I was thinking, well, the WBCO protein that's present in Pseudomonas aeruginosa might make a very interesting target for these compounds, but clearly that's not the case. On the other hand, we knew that Bacillus was very susceptible to inhibition by the tunicomycins, so this is 0.5 micrograms per well, by the way. But we were very surprised to find that the quinavosamycins were active on both the Bacillus and also on the eukaryotic host as well. So the surprising thing here is that quinavosamycins are active on a eukaryotic yeast. Okay, in summary, we found 12 genes that are essential for tunicomycin biosynthesis, including TUN-B, which is a radical SAM protein, and the TUN-D, which is this 111 anomeric to anomeric glycosyl transferase. Genomic library search of a actinomycetes genomic library using the TUN-B identified 4 new strains, tunicomycin-producing strains. I told you about one of them today. Streptomyces niger, the one which I just told you about, produces tunicomycins, which we were happy to see, and it also produces quinavosamycins. We've also cloned the TUN genes from that strain as well, by the way, and heterologously expressed them, and I hope to tell you about that soon. The quinavosamycins have a somewhat unexpected activity against a eukaryotic microorganism that we looked at, and I think that's a very interesting finding. I hope you'll find that interesting. Finally, the tunicomycins, we still believe these have analogs of the tunicomycins, and we've tested a number in my lab now, have the potential for MRAY-directed antibiotics based upon the structure of the tunicomycins. So just to acknowledge all the people who've helped with this over the years, and more recently, a number of people. Lin Shu was a postdoc of mine, and Biljana was a postdoc as well. They're now moved on to different places. Scott Kennedy is at the University of Rochester. My former colleagues, Frank Monomy, my present colleague, Mike Appel. Recent project that we've been working on, Wenqing Chen has been wonderful in Wuhan in helping us to clone the biosynthetic genes for these compounds. That's work that's now being done by Todd Naumann in the lab in Peoria. Mike Bowman, Todd Naumann, David Lebeda in Peoria, Ken Bischoff, who did the microbial testing, and Zixik Deng, who heads up the antimicrobial lab in Shanghai. And I'd just like to thank all of you and to ask for questions, please. Come on, folks, questions, please. Joe.
Yeah, so I suppose you were just mentioning some of these might become antibiotics. It would be great. I mean, it's synthetically difficult to make, so I assume you're going to use the gene clusters to try to actually produce analogs.
Right. So the question was about analogs and bioactivity of analogs. And so the synthesis is complicated. It's been done, in fact, by 3 different laboratories. We have some analogs that were produced in China by Bao Lu's lab in China, and we've tested those. It's work that's going on right now, actually. The interesting thing is to see if we can make specific compounds that hit specific translocases. So the obvious one that's of interest to us is the MRAY, because that's always the potential for antibiotic activity, if that's the case. We were a little surprised by this eukaryotic activity, by the compound that we've identified here. I thought the glutamate transferase involved in protein N-glycosylation was highly specific. In fact, I'd like to throw that open as a question to you. You know, is that actually the case? Because the compounds that had the quinnac head group were as active, possibly more active. We haven't yet done the broth dilution assays, but as active as the tunicomycins were. And to answer Joe's question, so we have cloned the genes for the quinavosamycin biosynthesis as well. It's the same operon as the tunicomycin. So the core genes that are involved in tunicomycin biosynthesis and quinavosamycin synthesis are the same core genes, but there are genes outside of the operon which are unique to the Streptomyces niger strain. And we're very excited about that, of course.
Because I'm not sure about the specificity of the oxacyclyl transferase or whatever you're talking, because of course you can use that as an antibiotic.
I agree.
But what what changes do you think might be
So
made?
one thing we found out recently, Joe, which was also kind of a surprise to me, and that is if you have the 2-azido sugar, if you take the glucosamine residue and you have an azido in that position instead of the amino group, that also is active. Okay, so that was a little surprising. I was of the belief that all N-linked glycoproteins all begin with a glutnac residue. So you have glutnac, then high man, and such on there. Sometimes a fucosyl group, but basically that's always a glutnac, and the dolichol glutnac transferase is a dolichol glutnac transferase. So there are two ways to interpret the data on the microbial plates, I think, and that is that, well, it's less specific than we all thought, and that obviously is going to take a little convincing. Or perhaps we're hitting something which is a different enzyme. It's always possible. Perhaps the tunicamycin is being lethal against the yeast in a way that no one was actually expecting, and that the quinavosamycins are also hitting this same target. Now, for example, tunicamycins also block protein palmitylation. Okay, so the RAS protein, for example, is palmitylated. The enzyme, which is the protein palmitoyl transferase, is also a target for the tunicamycins. Okay, so do we know whether that's the case for the quinavosamycins? And my answer to that is no, I do not yet know.
Myron doesn't have a question.
Oh, these are always the hard ones.
You're pushing the envelope here. This particular analog that you synthesize, have you did more studies on the eukaryotic cells in terms of the glycosylation and in terms of some of the receptors that are translocated to the membrane? Have you used that in order to block that? Because tunicamycin has been used at.
Right. Tunicamycin has been cited over 7,000 times, Myron. It's astonishing. It really is sort of like everybody's favorite tool. And the reason it's everyone's favorite tool is it hits a specific step, so it's very useful as a tool. So at the beginning of this work, and it would be untrue to say that. At the beginning of this work, I thought I discovered another strain that makes tunicamycin, which would have been sort of fairly interesting. What we found afterwards, when we found that there was a quinac residue there, we thought perhaps we had found a selective inhibitor for the WBCO family. Okay, now that was the fucnac 1-phosphate transferases that I mentioned at the beginning, where the recognition site was different to the muonex and the WEC-As. Okay. The reason that is important is that Yersinia pestis, the thing that causes Black Death, actually has a WBCO homologue that actually was the work that Matt Anderson and I originally did back in the depths of time when I think we probably had Black Plague. When we originally did that work, we actually used a Yersinia pestis strain in Merck. Okay, so the idea that there was a selective inhibitor for that would have been a rather attractive finding. Okay, so one of the reasons that I tested Pseudomonas aeruginosa here is that we thought that we know that the Pseudomonas has a homologue for that gene, and the hypothesis, all good hypotheses burn down in flames when you do get the data. So the data says no. The data says the quinavosamines do not touch the Pseudomonas aeruginosa. But so far, all we've done on that, Myron, is we've only done the microbial testing. We've not looked at any assays yet, enzyme assays, no.
Anything else?
All right, thank you.